Good afternoon, everybody. I am Lluís Alceda. I am the director of the Centre de Recerca Matemàtica, and it is my pleasure to welcome you here and to introduce our speaker for this colloquium today. The speaker is Roderic Guigó, and I am very happy that he is willing to explain the interaction between mathematics and applications of mathematics here. So let me explain a little bit who is Roderic, and then I will make maybe a personal comment about why I am personally very satisfied about this colloquium. Roderic obtained his PhD in the Universitat of Barcelona in 1988. He worked initially at the Department of Statistics in the development of mathematical and computer models in population genetics and evolutionary ecology. Then he joined the molecular biology computer research resource at the Dana-Farber Cancer Institute in Harvard, in biostatistics, in the biostatistics division, where he was a postdoctoral fellow until more or less 1991, that he moved to the Biomolecular Engineering Research Center at Boston University. During these years, he was involved in several projects in the field of sequence analysis, gene identification, automatic knowledge extraction from biosequence databases, protein sequence pattern analysis, and molecular evolutions. In 1992, he moved to Los Alamos, where he was a postdoctoral fellow at the Theoretical Biology and Biophysics group with James Fickett. And Los Alamos, he worked on genome analysis and related problems, estimation of genome's protein coding density, and characterization of large-scale genome structure. In 1994, he joined the IMIM here in Barcelona, and from 1994 to 1999, he was associated professor at the Universitat de Barcelona, and since 1999, he was at the Universitat Pompeu Fabra. In 2005, he joined the Centre de Regulació Genòmica, where he is working now, where he coordinates the Bioinformatics and Genomics Program. He's also a professor of bioinformatics at the Universitat Pompeu Fabra. He's author of about 180 publications, more than 180 publications, and he's a leading scientist in the field of computational genomics. He has other merits, like he's a member of advisory boards of many bioinformatics and genomics institutions and others. Taken literally from the web page of his laboratory, there it is written that his group is working in understanding the information encoded in genomic sequence and how this information is processed in the pathway leading from DNA to protein sequence. And they say that they are intrigued by the parallelism between computation and the processing of information encoded in biological sequence. I could continue, but I will stop here reading things. Maybe I should apologize for being so literal, but for me it's important to transmit you this, I think, unique way of focused about the interaction between modeling. Instead of talking about mathematics, I will talk about modeling and genomics and applied mathematics that you can find in CRG. I forgot to say that Roderic was collaborating in this, in all these things that I was saying in the finding, in the coding of the human genome. He had a very important contribution there. I read all this because I wanted to give the image about him, that he's a person that is sitting in a privileged area between mathematics, between genomics, between applications of mathematics, as I said, modeling. And I think that his sight on interesting problems is relevant and will be highly enlightening for us. And personally, I am very happy to have him here. He has been my contact as a director of the center in CRG. We are having CRM and CRG. We are having a joint endeavor in building research groups that will collaborate in doing research in this area, that they mix mathematicians and, say, biologists or bioinformaticians. And I hope that there will be a great progress in these joint efforts, putting modeling and bioinformatics together. And I'm very, very happy that we managed to reach agreements, starting with discussing with him. So I'm very interested in listening what he will tell us about, and I hope that we all will enjoy very much this colloquium. So, Roderic, you have the word. Thank you very much for being here.
Well, thanks Luis, for the invitation. And I'm really afraid, and maybe I hope I don't disappoint you after the introduction by Luis. I heard the name mathematics associated to my name, and this terrifies me because I am a biologist. I'm not a mathematician. I consider myself a biologist. And I will talk of really a small corner of the relationship between biology and, I would say, computation more than mathematics. I understand that, so this is the title of my talk, and I really sort of apologize to you because you are mathematicians and you like the things, the terms be very precise and very well defined. And actually, I will start by saying that neither the concept of life nor the concept of a computation are really clear. So we are talking of something that you are not used to, right? So it's also, I know that many mathematicians don't like data. I will start with data slide. It's this slide. That's a very simple search that I did many years ago in Google, but the results are more or less the same, in which I searched for terms in which I had a prefix that referred to a particular discipline, and then the suffix informatics. And I think that the slide is very striking. Bioinformatics dominates by orders of magnitude any other discipline in which you mix a scientific discipline and informatics. So by orders of magnitude. And this is often attributed to the fact, and this is actually the more striking because this discipline in bioinformatics did not exist until essentially mid-90s. Right? This is the number of articles in which the term bioinformatics appears. And this is, as I said, the success of bioinformatics is usually attributed to the fact that modern research in biology generates a lot of data, mostly because experiments have been automated. They can be carried out by robots, and there are many automatic ways of tracking and recording data on natural phenomena. And there was a paper, this published 10 years ago, that predicted that in 2025, genomics or biology would be one of the domains of big data. And it is true that biology generates a lot of data, but when I look at this table, I'm really not sure that biology generates more data than astronomy or than sociology or than meteorology, for instance. I don't know, maybe not. The hypothesis in which I'm going to base my talk, which actually does, whether it is true or not is quite irrelevant, is that this peculiar relationship between biology and computation has to do not only because of the amount of data, but because of the peculiar nature of biological data. So I will start trying to define what is life. As I said, this is not an easy problem, and it's not an easy concept to define. I will do it in steps. I will do some sort of iterative definition of what is life. So the first approximation, life, and when I talk about life, I mean life on Earth, is a DNA molecule with the capacity to produce a copy of itself. This is a DNA molecule. It's a string of 4 characters, A, C, G, and T, repeated multiple times. Of course, this is not a DNA molecule. This is a symbolic representation of a DNA molecule. Each of these characters corresponds to a chemical. These are the chemicals that are represented by these characters: adenine, thymine, guanine, cytosine. These are called nucleotides or bases, whatever, and they are conveniently represented by the initials of their names. So these chemicals generate polymers, that is, repetitions multiple times of combinations of these chemicals or of these letters. I will use letters, chemicals, nucleotides, and bases indistinctly. I know that that's not very precise for mathematicians, but that's what I will do. So they actually organize as a linear molecule with essentially a series of these molecules that are very conveniently represented by a string of characters. This is a fragment of what's called the human genome. I will define what the genome later on. Essentially, in our cells, the total DNA has a length of 3 billion nucleotides, 3,000 million letters, or depending on how you count 6,000 million letters, 6 billion letters. This is a little bit of a simplification of what a DNA molecule is, because the DNA molecule is not one single string. It's actually 2 strings, which is called 2 strands, we say in technical words in DNA, because as you realize, I presented these pairs of chemicals in groups of 2, because they actually chemically interact. Adenine interacts with thymine, and guanine interacts with cytosine. So what a DNA molecule is 2 of such polymeric molecules in an anti-parallel orientation, such that every time that I have every time that I have an A in one molecule, I have a T in the other molecule, and when I have a G in one molecule, I have a C in the other one. So the truly correct representation of a DNA molecule will be something like that. But we don't use this because, as you guess, once we have one strand, we unequivocally know which is the other strand of the DNA molecule. The fact that these 2 strands interact confers a stability to this molecule that is not a planar molecule. Actually, in space, it organizes, as you probably know, as a double helix. This is the discovery by Watson, by Crick, by Franklin, by Wilkinson, and others in the mid-50s. And this is the paper in which actually Watson and Crick proposed this structure of double helix for the nature of the chromosome, of the genetic material or hereditary material. This was, if I had to, I don't know which would be the paper in mathematics, I don't know which would be the paper in physics, maybe some of the Einstein's papers. I would say if I have to select one paper in biology from the last century, I would select this. And this is just a one-page paper in the Nature, in the journal Nature. And remember that I said, my definition of life, that life, it's a DNA molecule that produces a copy of itself. And there is a famous sentence at the end of this article. At the end of the article, Watson and Crick, they did not produce any model of how this copy was carried out, but he says, they write, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And indeed, the copying mechanism is very easy, it's very straightforward. This is a representation of a DNA molecule. I now use a different color for each strand, and the way in which this molecule produces a copy of itself is very simple. The DNA molecule opens, right? Opens, and then each of the 2 strands serves as a template to generate what is called the complementary strand, the auto strand. So when I have an A in one of these 2 strands, I incorporate a T. When I have a C, I incorporate a G, etc., etc., etc. And in this way, I go from one molecule of DNA to two identical copies of these molecules of DNA. This is a simplified representation of the molecule of DNA that's very convenient in some cases. Each of the 2 strands is in a different color, and the process by means of which the DNA replicates and produces 2 copies of itself is called DNA replication. So this is not a spontaneous process. This is a process that needs someone to carry it. The molecule that carries this process is very conveniently named DNA polymerase. It's a molecule that polymerizes the DNA. This DNA polymerase is a protein. It's a protein. This is this protein. This is the three-dimensional vision of this protein. Proteins are also polymeric molecules made from the repetition multiple times of a finite number of elements. In this case, 20 elements that we name amino acids. So, but the difference with DNA is that while the DNA always have, irrespective of the sequence, always have the double structure, the double helix structure, proteins fold in space depending on their characteristic amino acid sequence. Depending on the sequence of amino acids, these proteins acquire a different fold, a different structure in space from which the function of these proteins depend. So these are here you can see the 20 amino acids, and again, each of the amino acids can be represented, as in the case of the nucleotides, by a letter, by a character, and therefore the amino acid sequence of a protein can also be represented by a string in an alphabet of 20 amino acids. Okay? So we have now a system which is made of a DNA molecule and of a protein that can make a copy of this DNA molecule. These two elements are not independent. On Earth, these two elements are not independent. They are not floating in the environment and they met from time to time to produce the copy of the DNA. They're actually encapsulated. Well, sorry, I skip a slide. So we can, with this, we can have actually a further approximation to the definition of life. So life on Earth is a DNA molecule with a capacity to produce a copy of itself, plus a machine that produces the copy. What I was saying is that these two elements, right, the DNA and the machine that produces the copy of the DNA, are not independent, are not floating in the environment. They are encapsulated within a membrane that separates these two elements from the rest of the world. The system that's made from this membrane, the DNA and the protein that copies the DNA, we call it the cell. And I think this is important because on Earth, I emphasize this, on Earth we associate life to individual entities, right? We know there is a dog, there is a cat, there is a tree, right? There is a cell, there is a bird. So life is on Earth associated to individuals, to entities that are separated from the environment. So living beings are not clouds, right, in the sky that emerge, disappear. Or they are not like sand beaches that are continuously renovated. They are entities that had, this is very important, a permanence on time, during time. This underlines the concept of self, right? Of selves versus foreign self, myself with respect to what I am not, the self with respect to the foreign, which is very important in biology, in particular in areas like immunology, right, in which we need to recognize those things that are contrary to our interests, in a sense. So then we have a system that it's able to produce a copy of itself, in principle. But I guess that you may anticipate a problem with this definition of life. So we have this in one cell. We want to produce two copies of this. The DNA polymerase produces a copy of the DNA, and then the cell divides. The problem is I have two copies of the DNA, but I have only one copy of the protein of the DNA polymerase. How can I get the other copy of the DNA polymerase? And I think that this is the key thing of life, right? This is the key. I mean, maybe there is one thing that is to be clear is that actually what happens is that the DNA sequence contains the instructions to generate the DNA polymerase. So in other words, what happens is that the nucleotide sequence of the DNA specifies the amino acid sequence of the DNA polymerase, right? And I will go over this because I really think that this is what makes life a unique phenomena, different from the rest of natural phenomena on Earth. So, a better approximation to what life is life is a DNA molecule with the capacity to produce a copy of itself, plus a protein that produces the copy, such that the instructions to build the protein are encoded in the DNA sequence. And the way in which the DNA sequence encodes the protein sequence is not particularly complex. It's quite simple. It's a little bit unnecessarily complex because rather than this codification being done in one step, as it could be, it's done in 2 steps. I really don't know why this is. I don't know if anybody knows. So in the first step from the DNA sequence, and another nucleic acid, the DNA is called a nucleic acid, another different nucleic acid is generated, which is called RNA. And RNA is also a polymer made of the repetition of multiple times of 4 nucleotides, 3 of which are the same that those in DNA, and there is one, instead of a T, we have a U. We have a U. Instead of thymidine, we have uracil. This little change has an impact on the stability of the double helices produced in RNA. They are not very stable, and RNA molecules, in contrast to DNA molecules, are single-stranded polymers, right? They're just linear, single-stranded polymers. They do not complex with the complementary molecule to produce a double strand. So the way in which the DNA specifies the RNA is extremely simple. It's using the rules of complementarity. When the DNA opens, and one of the 2 strands, and this is important, only one of the 2 strands is used as a template to generate the RNA molecule. So when I have an A, we have a T. When we have a C, we have a C. When we have an A, we have an U. Now we don't have a T, and so on. So by this process, we generate the RNA molecule that's encoded into this DNA sequence. This is a schematic representation, right? We have the DNA molecule here. We have the RNA molecule. This process is called transcription, as I said, transcription, and is actually carried out by another protein called RNA polymerase, protein that produces polymers of RNA. So this is this protein, as you can see here. This is the protein, the RNA. This is the RNA polymerase. This is actually in the action of transcribing DNA to RNA. This is the DNA, and this is the nascent RNA, and this is the amino acid sequence of this protein. So this is the first step from the DNA to generate the RNA, and in the second step, we produce the protein sequence we wanted from this intermediate RNA molecule. This is called the translation of RNA to protein. And it's also a relatively simple process as well. It's a little bit more complicated because in this case, there is no one-to-one correspondence between the letters in the RNA code and in the protein code. That's impossible because we have 4 letters in the RNA code and we have 20 letters in the amino acid code. It cannot be a one-to-one correspondence. You need at least 3 letters in the RNA code to produce one amino acid. And this is what happens every three nucleotides determine a unique amino acid. And this is quite precise. This is according to two rules that are well established, that are essentially identical on all live beings. And what we go into this table, and this ACU, I go ACU, ACU codes for threonine, the amino acid threonine, which is represented by a T. I write a T, then I go to GUA, GUA codes for valine, and then valine is incorporated, and then following the rules in this table, the amino acid sequence of the protein is generated. Right? This table of equivalences between triplets, which are called codons, but doesn't really matter, and amino acids is the genetic code. Right? It's called the genetic code, and this genetic code is quite universal. There are exceptions, but it is quite universal from the most simple organisms to the most advanced organisms, whatever, if we are advanced or not, that's another thing. And again, of course, to produce this translation, right, to produce the amino acid sequence from the RNA sequence, you need something. And in this particular case, this something is called ribosome. It's not one protein, it's a complex of several proteins, doesn't really matter, and RNAs, but we can assume that's a single protein. By the way, this is a colloquium. I mean, if you have any question or anything, please interrupt me and ask questions, because I think it's important. I apologize because I know that this is high school biology and many of you have already known about this. But I want to emphasize, maybe I fail on this computational angle that all these processes have, right? So this is what I would say it's the origin of life on Earth. This appears on Earth about 3.7 or before 3.7 billion years ago. So it's essentially a membrane that contains a DNA molecule. It contains a protein that's able to produce a copy of the DNA, and then it has other proteins that are able to synthesize this protein, the DNA polymerase that's able to make the copy of the DNA. Of course, the DNA has to contain the instructions not only to generate the DNA polymerase, but also to generate the RNA polymerase and to generate the ribosome. So these instructions are encoded in different regions of the DNA molecule, which are called genes. Each of the things is a gene. And the entire DNA molecule in the genome, in the cell, is called, or the entire set of genes is called the genome. It's called the genome. Of course, there are not cells on the Earth today as simple as this. This is the simplest cell that has been so far described. And as you can see, the cell doesn't have only 3 genes, as the ones that I have described. It has 470 protein coding genes that include genes required for DNA replication, transcription, and translation, the basic mechanism of life, but also include many other genes that make life possible by interacting with the environment, synthesizing, for instance, the amino acids that are needed for the proteins, metabolizing the molecules that are required to produce energy for this process to be carried out, etc., etc. So this is in a schema of this gene. It has of the genome of this species, Mycoplasma genitalium. I did not mention the name. It has only 580,000 letters, nucleotides, and each of these things corresponds to a gene. Each of these genes, if the genes generates a different protein, and the proteins have many different shapes and functions. What I have described so far, this of which Mycoplasma genitalium is an example, is one of the 2 main types of cells that exist on Earth. It's what is called the prokaryotic cell. Our cells are not prokaryotic cells. Our cells are eukaryotic cells. Sometime, it says between 1.6 and 2.3 billion years ago, a type of cells a little bit more complex appear on Earth. And in these cells, there is a membrane, there is a different compartment that separates the process of replication of transcription from the process of translation. This compartment is called the nucleus, right, in which as I said, the transcription takes place, and the RNA gets exported to the exterior, which is called the cytosol, where the process of translation takes place. This is why sometimes this RNA is called messenger RNA, because it's the RNA that carries the information from the DNA to the proteins. And of course, in addition to these 3 genes or 4 genes that I mentioned, the ones that generate the DNA polymerase, the RNA polymerase, and the ribosome, there are many other genes in eukaryotic cells. Maybe the smallest eukaryotic cell has 3,000 genes or something like that, many, many genes, and these additional genes are the ones that are responsible for this amazing morphological diversity, and these are free-living eukaryotic cells. So let me, before moving into a little bit more of technical stuff, I just want to recapitulate a little bit what I have said so far, and if you have any questions, I'll be happy to answer them. Well, sorry, and what I also wanted to say before is that On Earth, there are about 2 million known eukaryotic species, and now there is a project that aims to obtain the genomes of all these species. So far we know about 0.1% of the species of the known eukaryotes, and the aim is in 10 years. I think this is very ambitious to obtain the genome of all these species. So this is a huge amount of data that will be generated. We don't know how many eukaryotic species exist on Earth. Maybe 7 million, maybe 8 million. There are a number of estimates. Many of these species will disappear before we know about them. So we will never be able to know that these species existed. We will not ever be able to sequence the genomes of these species. So in a sense, this is a race against the clock, because many of the species that we know will disappear within the next years. So the idea is to at least keep a digital register of all the species that we know exist on Earth. This is an international project that has many, many nodes, and there is a local node, actually, which is originated here in this institute, and it's called the Societat Catalana de Biologia, that aims to contribute to this project. It's called the Catalan Initiative for the Earth Biogenome Project. So now we have a quite good definition of life on Earth, I think. A living being is a system made of one or more DNA molecules, plus the molecular machines needed to produce the copy of this DNA molecule, such that the instructions to build the machines are encoded in the DNA molecules. And I know that I am not really making myself clear of how important or how unique this definition, how unique life on Earth is this idea that you have the instructions to produce something that is able to copy yourself. These things that are self-referential, sometimes that are in principle obvious, are sometimes difficult to understand. Maybe a good example is a quine. I don't know if you know what quines are. Quines are programs that, when executed, produce themselves. Right? This is a Python example. If you run this program, it produces exactly this output. So life is like a quine, but more complex. It's not only that it's a program that produces itself. It's a program that has the instructions to build the computer that interprets the instructions to produce itself. I like to bring this quote because I think, again, I really want to be able to transmit this message. No? This is a book that was written by Erwin Schrödinger, which was, I think, one of the most important physicists of the 20th century. And he wrote the book in the 40s because he said that all the problems of physics had already been solved, and really the important problems were in biology. And he was not a biologist, but maybe he is the most intelligent biologist of all times. Because before, this was written 10 years before Watson and Crick discovered the structure of the DNA, right? And he wrote this and many other amazing insights. So he says, the chromosome structures are at the same time instrumental in bringing about the development they foreshadow. They say they are low-code and executive power, or to use another simile, they are architect's plan and builder's craft in one, right? So this is the idea that you have the plans to build something, but at the same time you are the thing that builds something. Right? So again, I'm not sure I am transmitting what I want, but I think that this is quite. I think that this is quite amazing. That Erwin Schrödinger wrote this in 1944. He had a great understanding of biology. Well, I guess that if you understand quantum physics, you can understand anything, right? I guess that's probably the case. Anyway, just another trial to try to transmit the message that I want, and I will stop here with this philosophy thing. This is a sentence from a book that's called The Society of Spectacle. This is by Guigo, that's a philosopher from the mid-60s, well, not from the mid-60s. This belongs to a movement called situationists that were popular. I mean, only a few of people in this room will understand what I'm saying. They were associated to the May of '68 in Paris. So this is many years ago. I mean, I don't really know very much about this, but this is in this book. It is a criticism of capitalism, saying that under capitalism, we are becoming spectators or actors of our own lives. But what this sentence says, if you change spectacle for life, reads in this particular way. The tautological character of life stems from the fact that its means and ends are identical. And this is exactly what happens with life, right? In life, means and ends are the same thing, and in fact, he claims that this is a tautology, and you are mathematicians, you understand better than my sense that the tautology is. But in fact, there are many people that criticize the only theory that we have in biology, which is the theory of evolution by natural selection, as being a tautology, right? So I will not, the natural selection has been criticized as a tautology. So I will not go into this area. It's a philosophy I don't really know about this, but I thought that I wanted just to make yourself understand something that's at the time simple and complicated. I prefer to, because this is more relevant to what the discipline of bioinformatics is this idea of life as computation, right? This is the pathway that goes, this is something I work, it's the pathway that goes from DNA to proteins, right? This is what is known as the central dogma of molecular biology. This was proposed in 1958 by Francis Crick, one of the multiple discoverers of the structure of the DNA, which wrote the central dogma of molecular biology deals with the detailed residue by residual transfer of sequential information. And in fact, this is what we have seen. The information of DNA to DNA is the replication, the transfer information from DNA to RNA is the transcription, and the transfer of the information from RNA to protein is the translation. And Francis Crick postulated that there could be other potential ways of transferring information from RNA to RNA, from RNA to DNA, and from DNA to protein. So we know now that this thing exists. These are our friends, the viruses. This is SARS-CoV-2, and this is HIV. There is no, as far as we know, as far as I know, there is no transferring information from DNA to protein. And since I mentioned viruses, and I said that life is not very clear, I use actually the example of the viruses to reflect on this. So many people believe that viruses are not living beings. I am inclined to believe that they are living beings, but actually they do not match my definition of life. Remember, my definition of life is a DNA molecule, or in the case of the RNA viruses, an RNA molecule that encodes the instructions for the proteins to make a copy of itself. The genome of the viruses do not contain the instructions to generate all the proteins that are needed to copy the genome of the viruses. The viruses are very efficient, very intelligent if you want. They hijack the molecules from other living beings to produce this. So they infect our cells and they use our DNA polymerase, our RNA polymerase, our ribosomes to make copies of themselves. So in a sense, they do not match my definition of life, but they may be living beings. So what I want to emphasize here is that this process of information from DNA to RNA to protein is a computation, are computations in almost a paradigmatic sense. So in the case of translation, what I have is a string in a particular alphabet as an input. I have an output that's in a string in a different alphabet, and then I have a program, the genetic code, that transforms one string into the other, quite precisely, by the way. So I cannot find a better example to describe what a computation is, right? This is a computation in the paradigmatic sense. And this reflection of life as a computation, I think it's important because the mathematics that we use to deal with this part of biology are a little bit different than the mathematics that we use to deal with quantities. So I like this sentence from this book by Alvis Brathwaite. It's actually a friend of mine who wrote this thing. And I mentioned to you why I think that this is important. For billions of years, all durable information present on Earth was almost exclusively recorded in DNA or other polymer molecules. Other polymer molecules are RNAs or proteins. So that's what Alvis Brathwaite wrote here. And then he said, but a few 100,000 years ago, this changed. Human language emerged, providing an alternative means for recording and transmitting large amounts of information from individual to individual and from generation to generation. So what Alvis Brathwaite is saying here is that there are only two. On Earth, there are only two systems that maintain information. One is the human language, which I believe mathematics is included in the human language, and the other is the DNA. And I thought that this was a little bit of an exaggeration, but I've been thinking for a while, and I think that most likely he is right. And maybe, although I don't know, this is the reason why these large language models has been so successful resolving some of the most important problems in bioinformatics, actually in biology. So one of the early problems in bioinformatics was the problem of predicting the structure of the protein based on the amino acid sequence of the protein. This is one of the first problems that was posted, has been attracted a lot of attention. There were every year there was a meeting, maybe there has been already 30 such meetings or more, I don't know, which was called CASP, which is Critical Assessment of Protein Structure Predictions or something like that. So this was a challenge. Early in the year, the groups that were going to produce the structure of the proteins, mostly through crystallization, released the sequence of these proteins, and they asked the groups working on predicting the structure of the proteins to produce predictions, structural predictions of these proteins, and then they release in these meetings the real structure of the proteins, and they evaluate the performance of the methods. So there was a lot of competition. Actually, for many years, the best method to predict the structure was a person that looked at the sequence and was predicting the structure. But this changed like 2 or 3 years ago because Google released this program, AlphaFold, that defeated any other method that had been developed during almost 30 or 40 years. And in a sense, the problem of predicting the structure of the protein from the sequence has already been totally solved. And just let me, I know maybe I'm out of it, but I think that, and what I think it's important in this thing is that, as I said, the mathematics that we use to deal with this area of biology, or if you want, this area of biology is more similar to linguistics than to other quantitative scientific disciplines. And again, I like to bring this quote from Erwin Schrödinger that I find quite fascinating, right? Because what he wrote was the following, which I think nobody would say this, right? Because we think that the world is dominated by the laws of physics, and he was a physicist. And he wrote, what I wish to make clear in this last chapter is, in short, that from all we have learned about the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to the ordinary laws of physics. Because indeed, many of these processes that occur at the basic level of biology, this processing of strings, do not depend on the entities that made these strings, right? That these strings correspond to, right? In a sense, are independent of what is the physical material that mates them. Anyway, now going back to, let's forget about philosophy, going back to the data. So why this? I mean, this is not irrelevant because actually these sequences, the sequences of the genes, the sequences of the proteins, the sequences of the DNA, contain very relevant information about the function of the genes. So essentially the function of a gene, the biological function, what this gene does, what this protein does, and I use the term gene and protein in a sort of interchangeable way, is encoded into the sequence, not only the function, but also the history of the sequences. This is a sequence, let's say, I don't know, I forgot to acknowledge where I took this slide from. This is the sequence of a protein, let's say the DNA polymerase, in many different species, right? And this that you see here is called a multiple alignment, multiple sequence alignment. So essentially, each column in this alignment corresponds to the equivalent position in this protein in the different species. And you see that there is some conservation. At the bottom, we have what is called the consensus sequence. Some positions are very conserved in capital letters, some are less conserved, whatever. In some cases, there are places in which some regions of the protein had been lost. In some other cases, well, this is a mistake because if this was a DNA polymerase, the sequence would exist. So this is not a DNA polymerase, whatever. Some species may have lost this particular protein. And again, in the sequence of this protein, the function of the gene is encoded. The function and also the history. For instance, these are many different species all across the eukaryotic lineage. And here we have us, Homo sapiens, and this is the chimpanzee. And the two sequences are identical, right? It is essentially, with this maybe mistake, actually. And then these are more similar to each other than to the other mammals. These are other mammals. And these are more similar between them than to the other vertebrates. These are vertebrates, and more similar between them than to the other metazoans, animals. These are, or more similar to the metazoans plus these are fungi, these are plants, and these are unicellular eukaryotes. So the closer to a species, the more similar the sequence of their genes. And we use this to reconstruct the phylogeny of the species. So the building of these alignments is extremely important. Deciphering the sequence of the amino acids of the proteins is extremely important to understand their function. So the first amino acid sequence of a protein was determined by Sanger, Frederick Sanger, also in Cambridge. By the way, Watson Crick and the others were also in Cambridge, essentially at the same time, right? This is in early 1950. And this is the amino acid sequence of the insulin from a cow. And this is the amino acid sequence, right? The paper actually was simply the sequence of the amino acid. So this method that was put together by Sanger was then used in other labs across the world, and by the mid-60s, there were already hundreds of amino acid sequences known. Then this person, from which I am including the picture here, which is Margaret Hoff, had an idea, a very simple idea, an obvious idea, which is collecting the sequences that were in many different papers in journals, right? They were printed in journals, so they went to the journals and copied by hand, because there were not computers at that time, and put all of them together. And this is very important, right? Because you know that when you have many individual instances of events and you collect all of them together, you can generalize about what is common to all these individual instances. So usually, if you talk to a bioinformatician, they will say that Margaret Hoff is the pioneer or the father or the mother of bioinformatics. And essentially, the thing that she did was writing these books. These books are very boring. Each page on the book is just the sequence of amino acids of a protein, nothing else, right? This is the sequence of amino acids of a protein. And again, these were printed papers. We are in the pre-computing eras, right? Mid-60s. Computers existed, of course, but they were there were very few universities that had computers at the time. Computers were first used for weapons research, and then they were used in banks and other companies, and then they went into the universities. But what she could do is actually build these alignments by hand, because I said there were not computers, and he found this result, right, that sequences that have a similar function, they have similar sequence, and that the similarity between the sequences reflect their evolutionary relationships. So in some cases, a position is lost, it's a deletion. In some cases, a position is inserted. I don't know, I don't see here. Maybe this is an insertion here, right? But in some other cases, there are changes in the amino acid sequence. So by looking at these alignments, Margaret de Hoff built what is called substitution matrices. This is a very simple idea. Essentially, this is a matrix that provides for each pair of amino acids a score, which is a score that's related on how likely it is to see these two amino acids in the same column of an alignment, right? Remember, this is an alignment. This is an alignment. So looking at many of these alignments, he was able to determine how often it is possible to see L and Q together in the same column of the alignment that correspond to the same position in the protein. And this is quite important, because this allows to formalize in mathematical terms, which I guess is what we like, a problem in biology. It's the problem of finding the optimal alignment between two sequences. And I may be exaggerating, but I think that this is the first time in which a mathematical development was triggered by a problem in biology. It's not the first time. Of course, the early work on Fisher, on the statistics of the early 20th century and all this, they were working on biological problems, but these biological problems could be compared to other problems in other areas, right? The thing is that they were developing and working on this. But this was a problem that arose specifically in the area of biology. There was no other problem like that, right? And what is this problem? The problem of the optimal alignment between two sequences. It's also a very simple problem to face. These guys, Apostolico and Giancarlo, stated it in a mathematical term like the transformation of a string in another string that minimizes the number of weighted edit operations, and they define these edit operations, but I'll say it in the more biological terms that I am used to. So we have these two strings, right? We have these two strings, and we want to build an alignment with these two strings. There are many different alignments. If we incorporate the possibility of insertions or deletions, the fact that amino acids can be inserted or deleted in the protein sequences, and we compute a score for this alignment. That's very simple to compute. We go to this matrix, and when we see, I don't know, for instance, a tryptophan aligned to a glycine, we put a -7. By the way, these negative numbers means that this tryptophan and a glycine are unlikely to be seen in the same column of the alignment. For positive scores, right, this means that really this histidine and arginine can occur with quite high likelihood in the same column of the alignment. So, and I arbitrarily set the penalty to align an amino acid to an insertion or deletion, what is called a gap, to -4. And then I compute the score. And in this case, the score is -1, and in this case, the score is 6. So this is a better alignment than this. So the problem that we formulate is the following. Among all possible alignments between these two sequences, how can I find the optimal alignment? And this is a simple problem. In principle, I just enumerate all the alignments, I compute all the scores, and then I take the one that has the highest score. The problem is that I cannot enumerate all potential alignments because there are many, many alignments, right? For instance, between two sequences of 120 amino acids, the number of potential alignments is 10 to the 100. There are no computers that are able to solve this problem. And 120 amino acids are quite short proteins. Proteins on average have 400, 500 amino acids. There are proteins that have thousands of amino acids. And by the way, there is a version of this problem that is more useful, which is actually what usually we use in biology, which is the optimal local alignment. In this particular case, what we want is to select the substrings from the two strings that we want to align, so that these two substrings have the maximum score in the alignment. This means I have these two sequences. This is the global alignment. So all the residues are aligned with all the residues. This is the local alignment. This means that given my scoring schema, I cannot extend this alignment in any of the two directions and increase the score. If I extend the alignment in these two directions, the score or the alignment will be lower than this one. And by the way, this problem is related to the distribution of the longest head run in a coin tossing sequence, which is a problem that was addressed by Erdos and Renian. Since I am in an audience that are mathematicians, I thought that maybe I should mention the name of some mathematician, and the easiest mathematician to mention is Erdos, because he published almost any area of mathematics, including in computational biology. So there are, as I said, there is no, yes?
The previous example is like a slight difference in the sense that in this local alignment, you can add pairs that are not aligned that would temporarily reduce the score, but you can have a local alignment in which you have, like let's say, 3 pairs that are aligned, like 3 bases that are aligned, one that is not, and then 3 more that are aligned.
Yeah.
And then this local alignment is
It's the two superstrings that maximize the score of the alignment. So it could be this or it could be more. But if I extend them, the score of the alignment decreases. So this problem was addressed during the 70s and the 80s. These are two of them, I guess. Well, anyway. So and the way in which this problem, the algorithm that was put together to obtain in a more efficient way the optimal alignment between two sequences, the enumerating of them, was based on dynamic programming, which is this recursive method of obtaining solutions to problems. Essentially, it's based on the idea that the optimal alignment between two superstrings starting in the first position, for instance, this GCAT and this GAT, just the score of the optimal alignment depends only on the scores of the substrings that are represented by the cells in the matrix. So to compute this number, I only need to know these 3 numbers. And therefore, I only need to make m times n computations, and this is the complexity of the algorithm. And then I can recover the alignment by backtracking this matrix. So in this particular case, if you have to sequence of 100 amino acids, the number of operations that I would need to do to solve the problem would be 10 to the 4, not 10 to the 100. So these methods are still very much used in molecular biology, in sequence analysis. But what happens is that the number of amino acid sequences or DNA sequences was growing. So the methods to sequence DNAs were invented in the middle of the 70s, also by Sanger in Cambridge and Maxam and Gilbert in Harvard. And therefore, the number of sequences that were known were increasing exponentially. So by the early 80s, the first databases that had these molecular sequences were established. The first such database was established, it was called GenBank, was established at the Los Alamos National Laboratory. I guess that many of you now are familiar with Los Alamos National Laboratory because you've seen this Oppenheimer movie, right? So some of you at least have seen this movie. But if you don't have seen this movie, the Los Alamos National Laboratory was the laboratory where the Manhattan Project took place. This was the project that led to the fabrication of the atomic bomb. And this is actually related, this is actually the reason why the GenBank in Los Alamos. Because after dropping the atomic bomb, the effects on human beings of the radiation were mediated by mutations in the DNA. And therefore, there was established a biological division in Los Alamos National Laboratory that had as aim to study DNA. And because there was this division in Los Alamos National Laboratory that was studying the DNA, the first database of DNA sequences was established in Los Alamos National Laboratory. So as the number of sequences grow and these databases become first electronic, that did not exist before, a problem that was recurrent in biology during the 90s and the first 2000s was, and still is very important, is when you have the sequence of an amino acid, the sequence of amino acids of a protein, you want to know which is the function of this protein, of this gene. And the way in which this is usually solved, not just through experimentation as well, but by taking the sequence and comparing it against all the known sequence and identifying the sequence that have some similarity over a given threshold with this particular sequence. From the analysis of the sequences, from which hopefully the function is known, we can predict the function of this amino acid sequence. To carry out this comparison, the dynamic programming algorithms that were invented in the 70s and the 80s are too expensive. They cannot be used because it takes too much to even though they are very efficient compared to the naive enumeration of all the alignments, they are too slow. They are too slow to be practical. Therefore, the heuristic methods were invented that produce much faster solutions, and they were based on hashing and indexing. Essentially, what you do is you have your database, you index all the k-mers, so for instance, all the sequences of 6 amino acids, you know where they occur, this is your dictionary or whatever, and then you have your query sequence, and then you very, very, very quickly identify where in your database these k-mers, whatever, appear, and from here you can accelerate the identification of sequences that are similar to your query sequence. So this is, as I said, I'm not exaggerating when I said that this was one of the most important methodologies used in biology during the 90s and the early 2000s. This is the most cited papers in these 2 decades, and all these papers are methodological, so they are methods that people use in the lab. These are methods that people use in the lab. The third most cited paper is Basic Local Alignment Search Tool, because essentially one of the most used methods in biology, not only in molecular biology, in biology, was the comparison of a sequence with other sequences. And actually I realized that the 5 most cited paper during this period was also a paper that had to do with analysis, with sequence analysis. Bugs is a computer, maybe some of the people that here have heard about bugs, computers. And this is still, and this is, and there are many examples, multiple examples of how important this has been in biology. For instance, Doolittle was carrying just a very simple thing. He was comparing all the sequences in the database against themselves, and he found this relationship. This is an alignment between these 2 proteins, between an oncogene, which is a gene that's involved in cancer, and a growth factor, that's a gene that's involved in the growth of cells. So illuminating the molecular mechanism by which cancer arises, which is an anomalous process of cell growth. And this is a relationship that had escaped all the researchers in the field. This is not a field that is not active. I'm not talking about the past. So these are 2 methods of hashing and of compressing and indexing that are extremely used in biology nowadays. And again, I will justify this later. This is the Burroughs-Wheeler transform. That's a method to compress strings. Again, the similarity between textual languages and DNA. Essentially, what this does is a transformation of the string that makes characters that are identical appear together, right? So this is very amenable to compression. When you have a series of the same letter, let's say A 70 times, you don't need to put 70 A's. You put A and 70, right? And then you compress the data. So this is a method that's used in most compression algorithms. And then there is the Ferragini-Manzini FM index That's a very fast way of finding substrings in compressed data, and I don't know how this thing works. Sorry, it's too much for me. But to understand why these things are so important today in biology, we need to move back to biology for the last time. And it has to do with the origin of multicellularity and cell differentiation. So far, what I said about life refers to individual cells, right? So all living beings that I show are individual cells, but we are not individual cells. We are multicellular organisms. Our bodies are made of many, 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 many cells. How do we go from a single cell? What is the relationship between the single cell organism and our cells? Well, same time, as I said, I think 600 million years ago, when a cell produced a copy of itself, rather than, for whatever reason, rather than separating from the other cell, became associated to the initial cell. And therefore, these colonies of cells were forming. With the time, right, these colonies, initially maybe they disgregate, but with the time, these colonies of cells, right, grow and grow and grow until they separate part of these colonies to generate new colonies, right, to generate new colonies. So we could think about these agrupations of cells, right, as a proto-multicellular organism, as our ancestors. These are colonies of identical cells, and something that is very important, because these originate from the same cell, they all have the same DNA. They all have the same DNA. So this is said in another way. So we have colonies of identical cells, but with the time, some of the cells specialize. With the time, with evolutionary time, I mean, maybe 300 million, I don't know, 450 million years ago. And some of the cells have different functions within the colony than other cells. For instance, in this particular case, we have locomotor cells, cells that make the colony to move into a particular direction, and then other cells that are specialized in getting the nutrients from the environment that are needed for the survival of the cells, right? So this we can start considering this as an organism, not a colony, but an organism. An organism is a colony of cells that have some degree of specialization. And further later on time, there were a type of cells, right, called gametes, that exited the colony or the organism and were able to initiate a new colony with all types of with all cellular types in the original colony, right? These are called gametes. So these cells, at a particular point in the life of the colony or of the organism, whatever you want to call it, exit the colony, the organism, and are totipotent. They are able to generate, again, a colony that's similar to this one. Okay? There is something that I will not touch, I'm sorry, because this maybe attract your more attention, which is sexual reproduction, right? Because in sexual reproduction, you need two gametes to create an individual, but I will not enter into this. This also has very important so anyway, so this is the cells that we find, different types of cells that we find in our body. And maybe some of you, if you still follow me, may think, well, you're telling me that all the cells in this colony, in this organism, have the same DNA, but these cells are very different. If all the cells have the same DNA, means, I'm sorry, and again, so there is a project now. There's a project now that it's called the Human Cell Atlas, another big important project in biology that aims to define all cell types that exist in the human body. So these are only some of the cell types. We don't know how many cell types there are in the human body. We know how many cells there are in the human body. In each of our bodies, there are approximately 36 trillion cells in our bodies. Well, so the problem that I was trying to address, the issue that I was raising, is that there is all the cells in this multicellular organism have the same DNA. Therefore, right, they have the capacity to produce the same RNA, and therefore to produce the same proteins. The question is why the cells are not identical if they all have the same DNA. Well, the reason is the following. The reason is because not all genes work or are active or produce RNA in all cells. If this is my genome, some of the genes in red are active in some cells, for instance, in these cells. But in other cells, the set of genes that is active is a different set of genes. So through this combinatorial activation of genes, we can create the diversity of cells from the same set of genes from the same genome. And it is not only a binary, it's not only an on-off behavior of genes, it's more of a continuous, right? So maybe you don't see it well, but it's not only that the genes are on or off, but they can actually be on at different levels. For instance, the gene produces less copies of RNA than this gene, right? And the same here, the gene produces less copies of RNA than this, and therefore less copies of the corresponding protein. So through this mechanism, by means of which genes are activated or deactivated in different cells, again, from the same genome sequence, we can produce high cellular diversity. Again, this is very complicated for the point of view of biology. Biology is very complicated compared to mathematics, very, very, very complicated. So the way in which this thing is regulated is because there is a code, there's a transcriptional code, much more complicated and less well-defined than the genetic code, but essentially it's encoded in the sequence of the DNA, so particular combinations of letters or motifs, as I said, not totally conserved, right, appear in the sequence of the DNA. They bring together proteins that are called transcription factors that bind to the DNA, and when they are bound, there is the polymerase, the RNA polymerase, that initiates transcription, gets activated, and produces the RNA molecule, right? These combinations of these motifs that's different in different genes is what causes that different genes are expressed or work or are transcribed in different cell types. Therefore, the better way to define the state of a cell, the type of a cell, and the state means also the state in the case of disease, is by measuring, by identifying all the RNAs that are produced in the cell. And this is what biologists do, what we do. But the fact is that because of technological limitations, we don't have technologies that are able to obtain the sequence of the entire RNA molecules. The methods that we have require that we fragment the RNA and then we sequence these fragments. And then, to know which genes are active, we map these fragments that are sequences, we map these fragments that we call reads, we map them into the genome, and depending on the reads, the number of reads that map into the different genes, we know the level of the number of the abundance of RNAs that are produced by these genes. So typically, if you do have an idea, typically in one of these experiments, the sequencing of RNA, which are called RNA-seq, we produce between 10 and 200 million such reads of length between 75 and 200 base pairs. And to have an idea of the magnitude of the problem, of what the numbers that we are talking, I just asked the CRG, my institute, how many experiments were carried last year, and there were 22,000 experiments that on average produced 30 million reads per experiment. So this means that last year at the CRG, we produced 1 billion mappings of short reads into DNA sequences using these Urros-Weller and Ferrangini-Manzini methods, which are known to, I would say, 99% of the biologists, is the tool that they use more often in biology. This is a very active field of research, and I mean, if I go to the Wikipedia, this page is not updated since 2017. There are 60 different methods, probably there are hundreds of methods being developed to map reads into genomic sequences. and I am about to end, because because this is what allows me to link this, I don't know how to call this, more linguistic approach to the mathematics of biology to the quantitative biology that's probably the most dominant, is that when I get this mapping of the reads into the sequence of the DNA, from this I can infer the expression of the genes, which means the number of copies of the RNA that is produced. And these are already numbers. These are quantities, quantitative values that are amenable to quantitative analysis. So this is when quantitative biology at the molecular level arises. And this is very important, extremely important. This is identifying the genes that are expressed in different conditions and that are differentially expressed. It means that they produce different numbers of RNA molecules in different conditions. Is very important to understand the biology. For instance, these are genes that are more expressed in this form of leukemia. This is acute lymphoblastic leukemia, and these are genes that are more expressed, that produce more RNA in another form of leukemia, which is called acute myeloid leukemia. So this is very important, both in terms of diagnosis, these two forms of leukemia, and also in terms of treatment. And I asked ChatGPT, which are the areas of quantitative biology, and I just realized that what I've been talking to you until now essentially is a minor part of what is now known as quantitative biology, maybe part of what calls bioinformatics and genomics, but I think it's a unique part of biology because it speaks to the basic nature of what life is, right? Of what life is. And this is what really always have appealed to me. So I'm not sure I've been talking to you for more than an hour. I think that you are probably tired of biology. And so I think that I will skip this second part, but I just only brief you what is the second part is, because so you may think, well, this idea that life as a computation, yeah, is very interesting and blah, 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 but other than the metaphor, is this something that has any application? Well, I'll tell you that it does, because DNA can be used to compute. And this was first shown by Adleman in 94, I think. This was in 94, yes, in 1994, in this article called Molecular Computation of Solutions to Combinatorial Problems, in which he solved, I think that you are familiar with this problem, the travel-salesman problem. So the problem where you have a salesman needs to travel from one city to another, and he wants to know, or she wants to know, whether there is a path, right? And then there are connections by train or by plane. He wants to know whether it is possible, given these connections. Given these connections, there is a path that goes from A to Z, passing for all the cities only once, right? For instance, in this particular case of this simple, if the first city is Atlanta and the last city is Detroit, there is indeed such a path, I think, it's this, and this. But if the first city is Atlanta and the first city is Boston and the last city is Detroit, there is not a path that goes from Boston and Detroit passing only once for each of the cities. It's called the Hamiltonian path problem, which is to determine if a directed or undirected graph G contains a Hamiltonian path. That's a path that visits every vertex in the graph exactly once. This is if you have a graph of 4 nodes, you solve this by looking at the graph. If you have many nodes, it's impossible to solve. This problem is NP complete. And Adleman's generated an algorithm, which is not very obvious, right? The way in which Adleman, the way in which the algorithm of Adleman works seems particularly convoluted to try to solve this problem, but essentially the algorithm has the following steps: to generate random paths through the graph, then keep only the path at the start in the first city and in the second one, that have exactly the number of cities in the graph, and that have exactly each city in the graph only once. What is if this exists, the answer to the question whether the Hamiltonian path is yes. If it does not, it is not. I think the insight of Adelman was that he coded the graph using DNA molecules. So each city was in a string of DNA characters. It was longer than this. And then if there was a path from one city to another, right, there is a flight from Atlanta to Boston, this connection was also encoded by a DNA sequence that had the first part of this was the second part of the first city, and the second part of this was the first half of the city. He generated the complement to these DNA sequences that corresponds to the cities, and the sequences that correspond to the connections. There is a process called PCR, polymerase chain reaction. You put DNA molecules in some medium, and through this process, these molecules are copied millions, tens of millions, hundreds of millions of times. So in this soup, you have all the molecules that you generated. This is the molecule that corresponds to Atlanta. This is the molecule that corresponds to Atlanta to Boston. Because of the base pairing of the DNA, these two molecules get together, get attached, right? And then if in the soup there is another sequence that corresponds to Boston, to this, you will have it because, again, the base pairing, the double helix structure of the DNA, this will form, and so on. And if the sequence exists, at the end, this sequence is generated after you mix all this, then you know that there is a path that goes from Atlanta to Boston and that there is a Hamiltonian path in this graph. He did this using molecular biology because at that time sequencing was not as easy as now, but now you could sequence all these molecules and you will realize whether the sequence is in your medium, and then you could consider that there is a Hamiltonian path. So he says that he claims that this is much more efficient than computing and standard computers. So he said that in 1994, he could produce 10 to the 20 operations per second. Today, this is 30 years later, the fastest supercomputer at Oak Ridge Laboratory has a capacity of 1.2 exaflops, means 10 to the 18 operations per second, and it's very efficient energetically because with 1 joule of energy you can produce 2 times 10 to the 19 operations, when according to him, which is, I don't know, the theoretical maximum according to the second law of thermodynamics is 34 times 10 to the 19 operations per second. This was an academic exercise, but there has been progress in this area. It's called molecular computing. The method of Adelman to solve a particular problem. Computers are programmable. They can solve many problems. So there are now DNA computers that can solve multiple problems. And DNA can also be used to store information, videos, whatever thing that you want. So this was the first time, or one of the first times in which the DNA was used to encode. I think there were videos, there were back music, there were some text of Shakespeare, with a capacity of 2 petabytes per gram of DNA. More recent methods are able to increase the capacity of storage at 215 petabytes per gram of DNA, and there are even methods now for random access to the information. Now in computers, we access to the information in a way in which it's not sequential. So in the computer, we don't start with the beginning to where we look for a file. We don't start from the beginning until we find the file. Irrespective of when the file is, we have the same time of access. This is called random access because we have addresses that tell us where the information is in the computers. So I think that I have finished here because I think I took you a lot of your time. I don't think these final remarks are very important. I just, again, wanted to emphasize this peculiar relationship between DNA and computation. And I believe that because of this, computation is so relevant in biology. And thank you a lot. And sorry, I took maybe a lot of time. I hope that